From the research of polio to COVID-19 vaccines, these discoveries can all be traced to one woman. But you may be asking, how can a researcher or doctor be this influential? But the reality is that she was not a researcher nor a doctor. She was a woman whose cells were taken from her without consent and used to fuel these discoveries. In 1951, Henrietta Lacks visited Johns Hopkins Hospital in need of help for vaginal bleeding. She was diagnosed with cervical cancer and given radium to treat it. However, before her treatment, the cells were taken from her malignant tumor without her knowledge and sent to Dr. George Guy's lab, a prominent scientist in tissue culture. Henrietta Lacks's cells were unlike any other. Instead of dying quickly, they doubled every 20 to 24 hours. Dr. George Guy took this discovery and shared it all over the world. Unknowingly, Lacks was the reason for many influential discoveries, and although she passed in 1951, her cells are still alive today. The breakthrough in research using HeLa cells is actually because they are an immortal cell line. Cell lines typically divide until they eventually reach their end, or programmed cell death called apoptosis. But immortal cell lines can multiply to allow researchers to repeat experiments and reduce the amount of tissue samples needed. HeLa cells specifically allow George Guy and hundreds of other scientists conduct their research and make discoveries in things like polio and the Human Genome Project without interruptions. But because of the transformative nature of immortal cell lines, scientists have learned how to immortalize non-cancerous cells as well. One way scientists can immortalize cells is by targeting telomeres, which are the ends of chromosomes. In normal cells, the telomeres eventually shorten, resulting in cell death. Researchers can insert cDNA coding for the protein called hTERT, which is human telomerase reverse transcriptase. This causes the telomeres to not shorten, and thus cellular senescence is repressed and the cell can proliferate indefinitely. Another way scientists can immortalize cells is by targeting proto-oncogenes. In normal cells, the proto-oncogene works to regulate cell growth by stimulating cell division. But scientists can induce cell immortalization by inserting a plasmid that mutates cell division. These are called viral oncogenes. Oncogenes work as an accelerator or green light. Instead of regulating normal cell growth, they press on the gas for cell growth even when a mutation is found. Overall, the insertion of hTERT and oncogenes can cause cells to mimic the uncontrolled growth of cancerous cells. By creating artificial mutations, scientists can study cells with similar phenotypes and genotypes. However, when studying cells that come from human samples, it is important to understand the ethical implications, because even though HeLa cells have made a lasting contribution, we cannot ignore the injustice done to Henrietta.